Okay, thank you for the introduction, Volker, and for hosting this today. So as Volker mentioned, I'll talk in some sections. So first we're going to talk about an expression analysis, how to do that in JMP Genomics, kind of what your goal is, and then we'll take a little break and you can ask questions related to that topic. The second part, we'll talk about a genetic example. You might think of this sort of as a GWAS. There are a number of types of analysis we can do with genetic data, and so I'll take you through a couple examples of that, sort of a case control or a mixed model, kind of an ANOVA example of genetic data. And then the third thing we'll look at is plant breeding. And when we're talking about plant breeding, I'm going to show you a little bit about progeny simulation, but you can't simulate those progenies unless you have some kind of a predictive model that's giving you those simulated phenotypes. So we're going to talk a little bit about predictive modeling in that last section also. The predictive modeling could apply to expression or genetic data. It's not specific to plant breeding, but that last example will be a plant breeding example where we're going to do progeny simulation using some predictive models. Okay, so I'm glad that you're all with me today, and I hope that I'm able to answer a lot of questions, but I'll start by giving you some food for thought, and hopefully that will generate some questions as well. So I'm going to start with some slides to kind of show you where we would be going with this analysis. This particular analysis is an expression analysis on breast cancer, and specifically estrogen-treated samples versus not estrogen-treated control samples. So the data set comes from the Gene Expression Omnibus website. So you can see that the link at the top, if you wanted to go and download these same data, you could do the same thing that I've done. The experiment was done on using Affymetrix, and we had estrogen treated and then untreated control cells, and we're going to look at how a number of markers are differentially expressed between those two groups. So we're looking at these particular tumor cells exposed to estrogen at 3 different time points: 12 hours, 24 hours, and 48 hours after exposure. So we're going to measure them at those times after exposure. Okay, when I'm on this website, the way I'm getting data, because I'm not a researcher myself, I'm a statistician who wants to analyze the data. I didn't actually collect the data myself. So I'm collecting this data by downloading it from this website. Very likely you, if you're attending this webinar, you probably are collecting your own data. So if you're not getting things that are already in sort of a standard format from a website, you may have data coming in from a large number of different possible platforms. JMP Genomics has a lot of tools to deal with a lot of different types of data. After I go through this example, I'll show you a couple of other types of data files that you might bring in. In this case, I have cell files, so I'm downloading this zipped pile of cell files. I'm going to have a folder full of cell files, and then I can use some tools in JMP Genomics to bring those in. Once I bring those in using an import tool in JMP Genomics, this is the data set that I get for the actual expression data. So you can see there's 54,675 rows. That's shown here in this rows panel on the left. That's how many probe sets or markers there are. So in this case, I have what's called a tall data set. I have the many markers going down as the rows, and I have across as the columns the different samples. So I have 18 samples, a total of 21 columns. There's a unit column at the beginning that's sort of an ID, probe set ID, which is a different ID, but essentially redundant. They're both sort of an ID variable. And at the very end, after the 18 samples, there's one extra variable that I'm not using as well. Related to the actual data, I also need something called an experimental design file in JMP Genomics. In order to be able to do this analysis, I need to tell the software what are the variables that what are the things that I varied. So I have the estrogen treatment versus the control. So that's under this characteristics column. I can mark which samples were the control and which samples were the estrogen treated. I also have a time variable, so I can mark whether the measurement was taken at 12 hours, 24 hours, or 48 hours. And those are pretty much the only experimental features in this particular design. So this is a pretty simple design. Pretty much the most complicated I'm going to get with the experimental design here is maybe a factorial of the time and the characteristics. So I could look at the effective time, the effect of treated versus control, and I could look at the effect of their interaction. So is there a different effect between treatment and control at 12 hours than there is at 24 hours? So having a data set called this experimental design file, or EDF. This is what's going to allow me to choose those factors and build the model later on in JMP Genomics. So I need these two data sets, one explaining the experimental design and that original one with the actual expression values in the data set for each probe set or marker. When I'm doing an expression analysis, there's a lot of things I might be interested in. First thing, of course, I'm going to import the data. And then very common expression analysis tools would be looking at the distribution of the samples and if they are wildly different, perhaps normalizing or standardizing. We have different techniques to do this for microarray data versus for RNA-seq data. Then we might want to look at something like a correlation and principal variance component analysis to confirm that the treatment versus control actually is explaining some features and to get an idea of what experimental design factors are important in the model. And then I might actually make a model. So an ANOVA is a pretty typical model for an expression analysis. We could choose other things instead of ANOVA. We could choose a survival model or even a predictive model if we wanted to try to build something with a classification or regression tree or more complicated models. But it's a pretty standard approach to look at an ANOVA, so that's what I'll show you today. And all of these steps that are in this blue section, there's a special pipeline in JMP Genomics. So I could do each of these pieces separately, or I could use what's called a workflow in JMP Genomics, which already pipelines all these pieces together, because it's very common for someone doing expression analysis to be interested in all of these steps. You can just click through one process to do all of these steps. That's called a workflow. Or you could pick and choose the components yourself and sort of make choices as you go along, and you get more flexibility if you choose to do them sort of a la carte, one piece at a time. But there's a nice workflow, and we're going to look at that today. And then in addition to those standard things that are in the workflow, we could also look at predictive modeling. We could look at hierarchical clustering, principal components analysis, cross-correlation, subgroup analysis. There's a number of other more deeper things, deeper statistical things that we could also do with expression data. This journal that you're seeing on the left side, it's titled Workflow Journal. This is the output from JMP Genomics when you run this workflow with all of these steps in it. Each of these gray boxes is a button. I'll open this as soon as we've gone through this example and I've shown you what our goal is. I'll actually show you what this journal looks like and how you interact with it. You would click on each of these buttons to generate the report that's sort of hiding behind them. So this is the workflow journal is a nice summary of everything that you just did. So once you set up the process, you hit run, it runs, and then it produces this workflow journal for you. It also produces a ton of other background files. So you may not open or click on many of the files that it creates, but they'll get referenced in this journal. So JMP Genomics is trying to make it easy for you to sort of process which pieces of the output are going to be important for you to view. So this journal is kind of a nice tool to help you find the important pieces. So we'll click on this in a little bit, but you can see the steps that we would have gone through in this workflow are first we would just look at the distribution of the expression for the various markers. Then we would do a correlation and principal variance components analysis based on the raw data. Then we might have a choice to standardize or normalize the data. So we can make a choice at that stage, and depending on our choice, then for step number 4, we see a different distribution analysis after normalization. And then we see a new correlation and principal variance components analysis after normalization. And then the last step is to actually run a model to see if we can find differentially expressed markers. This is what the normalization process looks like, and this is what the output looks like that you can explore. The actual example I have, those cell files that we downloaded from the Geo website, they were already normalized. So I'll show you the picture of this in a moment. There was no need to do a normalization. So I'm showing you a different example here. This example shows that the expression, which each of these curves is sort of representing, you'd kind of hope for a normal distribution, that lots of the probe sets or markers have high expression sort of in the middle, and then that we're sort of middle range expression, and that you'd expect a tail of some markers will have very high expression and another tail on the bottom end, some markers will have very low expression. So what you hope for is a similar picture between all of these. Each curve is a sample. So in our case, we have 18 samples. This is a different example, so there are more samples. I think this example had like 400 samples, something like that. So we're seeing a line for each sample. And I've colored the samples by control or treated. Again, it's a different study that we're looking at right here, but we could imagine for our estrogen treated, they would be one of these colors and the control would be the other color. So we can see if there is an effect on this distribution of the control versus estrogen. We can also start making up some ideas of how we could normalize these. So we want to normalize these data because we expect there to be things like differential fluorescence between different microarrays. There's lots of reasons for expression to get to be read differently between different samples. So normalizing, we're saying, when we decide to normalize, we're saying that we believe that probably some things will get overexpressed in some samples, some things will get underexpressed, but overall the general distribution is that everybody has approximately a similar amount of expression with a similar amount of overexpressed and a similar amount of underexpressed. So when we want to normalize, things we might do are to shift the middle of each curve so that we push all the medians to the same spot, or we push all the means to the same spot, or we might shrink something about the variance. We might look at a trimmed mean instead. So there are a number of options, again, different options for microarray than there are for RNA-seq. RNA-seq has more options because of the way the data are collected. So after normalization, you can see that the choice I made here, which I believe was I shifted them to their medians and then did something with the thickness of the bands as well. So you can see that this after normalization has the curves looking much more similar. So at this point, we're hoping that we've corrected for different fluorescence, different reads in the expression between the samples. The next step, so here's what our data look like. This was before or after normalization because they were already normalized. So they look pretty much quite similar. The samples are all on top of each other. There's a second view of that normalization as well. So we could look at them sort of as density curves here, or we could look at them as box plots. The box plots, you're hoping that that green line that's signifying the mean across all of them, you're hoping that that kind of lines up across all of the samples. And the samples are grouped within their total treatment. So this is the hour and the estrogen or control. So you're hoping that the means of the groups are similar, and you're hoping that the spread is similar and the medians are similar. And so we can see that this is each of these box plots looks very similar to the others. We also, before and after normalization, will get output on the correlation and principal variance components analysis. Here I'm looking at the 3D scatter plot and a scree plot. The 3D scatter plot is showing me that if I summarized all the differences that are in the data, all the variability in the data, if I summarize that with just a few dimensions, in this case 3, so I'm calling those principal components. Principal component 1 explains about 30% of the variability in all of that expression. Principal component 2 explains an additional 14% of the variability, and principal component 3 is chopped off the side, so I can't see the percentage of it. It's going to be a little bit less yet than principal component 2. I can also see the points are colored. Again, they're colored by treatment group, so I can see that whether you were treated with estrogen or not is making a difference on the variability in expression. So that's a nice confirmation for me that in fact there are differentially expressed genes to find. There's going to be something interesting here. The scree plot is just helping me to see visually how much extra percent of variation is explained every time I add another component. So I can see that with one component, I'm here at about 30%. I add another component that adds another 14% of variation explained, then another component. So I can see that if this had an elbow in it, if it sort of tailed off quickly, and so you can kind of see the elbow better with the regular proportions, not the cumulative. This kind of looks like 2 principal components explains a huge amount, and we get kind of diminishing returns after that. So that's just a way of thinking about how many dimensions you really need to look at to capture most of the data. We also can look at this correlation heat map, again, to see what sort of trends are just there in the data before we even fit a model to this. And this is really telling. You can see the blue on the left panel. The blue are the untreated controls, and the red lines on the bottom are the estrogen treated. The blue and red inside of this box is not related to this control and treated. The blue and red inside of this heat map is related to being over or under expressed. So this is a heat map. The red spots are the hot spots. So the red spots are overexpression or high correlation. And the blue spots are low correlation. And then it's grayish color in the middle is no correlation. So we can see that the red hot spots, the high correlation, estrogen at 12-hour, the 3 estrogen treated at 12-hour, they're all very highly correlated with each other. So that makes sense. They had the same treatment measured at the same time. But that estrogen treated at 12-hour is not as correlated with the other estrogen treated. So we can see that estrogen treated at 24 is also very highly correlated with themselves. Estrogen treated at 48, very highly correlated with themselves. But those two are also pretty correlated. 24 and 48 hours, they're still pretty correlated. So this suggests that maybe more of the effect of the estrogen treatment is happening after the 12-hour mark, because that's not as highly correlated with 24 and 48 hours. We can also see these big blue chunks that the untreated controls are very similar to each other, but very similar to each other here, but very different from the estrogen treated and the off diagonal. Okay, once we run the model, we're going to run an ANOVA in this case, and we're going to look at the time as one of the factors. We're going to look at the estrogen treated versus control as one of the factors, and then their interaction. So when we look at this, we can see comparisons of any group of those experimental conditions. So in time, for example, we could compare the 24-hour average to the 12-hour average. This is called a volcano plot, and each point in this plot is indicating one of those probe sets or markers. And it's a plot of, on the x-axis, how big that difference is in the expression at 24 hours versus 12 hours on average. So the farther the points are to the right of the plot, the more 24 hours is expressed compared to 12 hours. The farther the points are to the left of the plot, the more 12 hours is expressed compared to 24 hours. And the higher the points are in the plot, the more statistically significant they are. Anything above this red dotted line meets the cutoff that we're asking for when we run this model. We asked for a false discovery rate here as the p-value cutoff. So everything above this red line is statistically significant at this value. But we really care about the things that are both highly significant and have a bigger difference. So the things that are splaying out to the sides, to the top right and left corners, are the things that are most interesting. And we can see that we also have a plot for 48 hours versus 12 hours. So we see which markers are statistically significantly different in expression between 48 hours and 12 hours. Now, I took a screenshot of this and put this in this slide deck before really thinking about what this meant, but I actually think it's a kind of nice example. The comparison between 24 hours and 12 hours is actually not a good comparison in this data set, because I'm averaging all the 24-hour points over the control and the estrogen treated, and I'm averaging the 12-hour over control and estrogen treated. So just comparing the time without separating it out by estrogen or control is telling me nothing that's actually of interest in this data. I'm just seeing what's different in expression from 12 hours to 24 hours when I muddle all of the samples together, control and treated. So I actually want to choose carefully which differences I make comparisons of. So I'll show you a tool in a moment that helps us pick which differences are actually of interest. Here's a close-up of a more interesting difference, the estrogen treated versus the control. Because the estrogen treated is first in that difference, anything that's a positive value was higher expressed for estrogen. So everything on the positive side of this plot is a marker that had higher expression for the estrogen treated than for the control. Everything on the other side had lower expression for estrogen treated than the control. Here's the comparisons within an hour between estrogen and control. So this is the much more interesting differences when we take time into account. So at 12 hours, estrogen treated versus control. At 24 hours, estrogen treated versus control, and at 48 hours, estrogen treated versus control. And this difference chooser, this gives us all the comparisons we could ask for. So the time differences, which are actually uninteresting to us because they're averaging over control and estrogen. So I wouldn't want to include any of the time differences. I probably do want to include the overall comparison between the control and the estrogen treated, and I might want to look at the time by characteristic comparisons where they hold the same hour, but the control and estrogen switch. So I can just tick mark all the things I want to include, and if I want to switch the order that they're subtracted, I can click this reverse order. So it makes it pretty easy for me to pick just the interesting comparisons and save all of my power for those comparisons. Here are the rest of the comparisons that may be of interest to me where I'm looking within the control or within the estrogen at different time points. And of course, you can see that in a lot of these things where we would kind of hope there isn't a huge amount of statistically significant differences, we're actually seeing a lot. So this is just a reminder that when you do thousands upon thousands upon thousands upon thousands or millions of tests, you're going to get a lot of false positives, which is why we ask for things like a false discovery rate to try to control for that. Another thing that we're going to get in the output is this dendrogram, which is showing us which markers seem to have similar expression patterns. So each row across this, there's so many rows. There were 50,000 markers, I think. So there's 50,000 something rows here. So each row is one marker, and the markers are being grouped. So there's a blue group up here in this dendrogram. So this is a clustering algorithm, and it's saying these that got this blue tag on the right side, these have a similar expression pattern. And these that have this orangey color, these have a similar expression pattern. These that have a green have a similar expression pattern. If we look at the green, it looks like that expression pattern is that, so the blue is down-regulated and the red is up-regulated. So high expression for red, low expression for blue. So it looks like this greenish chunk. They have lower expression for the control compared to the estrogen treated. And then across the control for all the time points, so control for 24-hour, 12-hour, and 48-hour, they're listed in a strange order here. It's mostly blue for that whole segment. So the control at any of the time points was either moderate or lowly expressed. In comparison, the estrogen treated at 12-hour, the estrogen treated at 24-hour, the estrogen treated at 48-hour, those tend to be overexpressed, higher expression. And then here's the band for just the average of the estrogen treated over all 3 time points. So we can see that that's a similar expression pattern for those particular markers, that they tend to be underexpressed when they're control and overexpressed when they're estrogen treated. We see the opposite of that for this reddish group, overexpressed for control, underexpressed for estrogen treated. And then we see a parallel plot showing us, again, the same kind of concept. The yellow lines here are tied to this yellow group, so we can visually see the higher expression versus lower expression in these different groups. The last thing we might want to get out of this analysis is to actually see overlapping, which markers were important in overlapping different comparisons. So this is a diagram showing 24 hours versus 12, 48 versus 24, and 48 versus 12. And so there are 251 markers that were significant in all 3 of these time comparisons. Again, the time is not the interesting thing here, but I wanted to be able to show you something with 3 things to get an overlapping Venn diagram that looks interesting. So if we wanted to compare just any set of our comparisons and look at the overlap of which markers are significant in all of those or in some of those tests, then we can do this. Once we select which group or control click to select multiple sections of this, then we can choose the button Show Selected Rows, and we can actually open a new data set that we might take into another analysis, like a pathway analysis. So if we ask for those selected rows, here's the 251 rows that were significant in all of the time tests. If we had extra annotation information that we could have carried through this analysis with the data, we could now grab this data set with all the annotation information and we could upload it to some other pathway analysis software. So JMP has some linkages to other third-party pathway analysis software. Okay, so what we looked at was with expression data, we looked at normalization, exploring some of those sources of variation with that scatter plot. We looked at similar expression profiles with that heat map and the parallel plot, and then we actually did the ANOVA and looked at those volcano plots to find which markers were significantly differentially expressed. And we could use a Venn diagram to choose which things overlapped between tests and then maybe take that to a pathway analysis. So I'm going to pause here and just very quickly show you what this looks like in JMP. So I have JMP Genomics open on the side. Here's what the data sets look like. So this was that experimental design file. Here's the expression data. And when I run this in JMP, I would start over here with this workflow. I chose the basic expression workflow in this case. So I would choose basic and then basic expression workflow. And this is what it looks like to use JMP Genomics. If you need help, there's a process description at the top. If you click on that, it takes you to the help about this workflow. Another place to get help is at the bottom. There's a load button. If I click the load button, if there's an example in the software, it'll suggest it. So I'd say okay. And now it populates an example so I can see how to fill out these various tabs. So it looks a little bit overwhelming until you've looked through one example, but we've got a lot of documentation on this and a lot of examples, and I'll show you where you can get to some blogs on our community page that can be really helpful in seeing some examples. One thing I mentioned is there's a lot of normalization methods, a lot more for RNA-seq. So this is the options for microarray data, but there are a lot more normalization methods for RNA-seq data. So that's the workflow that says basic RNA-seq workflow. Once I've run that, then I get a folder of output that looks like this. It's kind of overwhelming the first time you look at it because it just dumps so many files in here. It dumps some SAS data procedure steps. It dumps some SAS data sets. It dumps a bunch of JSL scripts, so JMP scripts. So what's happening is that JMP Genomics is a hybrid of SAS and JMP. So it's doing a lot of things in SAS in the background, but you're seeing the output in JMP. So it's a whole bunch of a mix of files because it's doing a lot of things behind the scenes that you're never touching. But what I like to do is search on the type of file. And if I just sort them, the JMP journal is the thing that's summarizing everything I just did in that workflow. So this journal.jrn, this is the thing I want to open. When I open this, I see that distribution analysis. So if I click on that, I see here it is. It already looked normalized. I could look at the correlation and principal variance components analysis. This rotates if I want it to. I could look at it as 2D plots instead. So I can see more of the principal components this way. There's the scree plot, correlation heat map, and so on. There's even the SAS output if I wanted to see what those eigenvalues are. I don't find that particularly useful for myself. Then I can do the data standardization, which just shows me the new data set that's created. I can always click this open button if I want to see the new standardized data, but in this case there's no difference, so I won't click on the distribution and correlation again, but I'll click on the ANOVA. It's going to now pull from a whole bunch of data sets that are sitting here to recreate that report for me. So this is going to take maybe a minute as it grabs several different data sets and recompiles them into that interactive report. Okay, so this is what our output looks like from actually doing the ANOVA. So each one of these was a test, was an ANOVA, using the expression as the response and using the time and the estrogen versus control as the factors in that ANOVA. So each one of these points is a different test for a different marker. And I can see I've set the probe set ID as the label, so I can see which marker it is. I can see how big that difference was in expression. I can see what the p-value is, or a version of the p-value. This minus log 10 of a p-value is a helpful way of looking at tiny, tiny, tiny p-values, because it basically tells you how many decimal places there are, like how many 0.000001, how many of those zeros there are before you get to a number in that tiny, tiny p-value. So in this case, there's about 15. It's about 15 decimal places to get to the 1 for that p-value. So I can grab points, I can select points, and it will show me where those points fall in all the other plots. Everything is interactive, so those points here that I grabbed here, I can see that they're more spread out in this distribution of p-values and this distribution of p-values. So I could easily explore whether some of my highest p-values are also high p-values in the other comparisons. I can hover over them to see, again, what their labels are. And then I can do other follow-up things like a gene set enrichment. I can look at that Venn diagram. The Venn diagram says, okay, which comparisons do you want to make? I'll do the time comparisons again. And so here with time comparisons control to estrogen, there are 2,484 significant markers in common between all of these. So this is actually still quite a lot. Okay, so now is a great time to take questions. If there's anything you want to ask me about expression analysis, then we'll look very briefly at a GWAS type of study and then very briefly at a plant breeding example.
Thanks, Ruth. So there are a couple of questions, and two of them are about the product and
Sure.
how to use workflows. One is, does JMP Pro include the workflow journal functions, or is that unique to JMP Genomics?
Oh, good question. Yeah, so
maybe we can couple this with a second one, which is related, and this is, I'm currently using JMP 14, so to get JMP Genomics, do I have to order it separately? Or JMP 14 already has a JMP Genomics component, so this is about products and journals and workflows.
Right, great questions. So if you are using JMP right now, you might notice that this looks exactly like your JMP, except it has this extra genomics tab, and you're seeing this genomics sort of, I don't know, starter window down below. It is not the case that JMP Genomics comes automatically with JMP or JMP Pro. So if you own a copy of JMP or JMP Pro and you want JMP Genomics, that's a separate license. So you would need to get in touch. If you're an academic, you'd get in touch with me or Volker or someone else on our team, and we can give you information on how to get a license of JMP Genomics. If you're a commercial person, if you're working for a company, you can still send us an email and we'll get you connected to the right person to find out how you get a copy of JMP Genomics. If you have JMP Genomics, you will have standard JMP 14 along with it. So JMP Genomics does have JMP included in it. It also has some SAS components included in it. So if you buy JMP Genomics, you will have the standard version of JMP from JMP 14 version. Now we're coming out with JMP 15 in September of this year, and JMP Genomics is not on the same cycle, so it won't update to JMP 15 when we get JMP 15 this fall. So if you have JMP already and you want JMP Genomics, you can license JMP Genomics separately. If you have just JMP Genomics, you would also have JMP 14 along with it. But that's the standard JMP, not the Pro JMP. So if you have JMP Pro, that's got more features in it than are in the JMP that comes with JMP Genomics. If that wasn't incredibly clear, just send us an email and I can try to clarify that again. But they are different products that have some overlapping things between them.
Maybe one simple yes/no question.
Sure.
Can I do proteomics and metabolomics with?
Yeah, so proteomics, metabolomics, other things like that, anything where you have some kind of measure of whether it's expression or something related to a numeric quantity that you're measuring for the probe set or the marker or the gene or whatever you're looking at, you'd end up with a data set that looks a lot like this. You'd have samples and you'd have your markers and you'd have whatever you're measuring. So if it's metabolites, you'd be measuring the amount of the metabolite. So you would actually use a lot of the same tools for expression for metabolomics and proteomics. You could run the same workflows for those. There are also a few extra tools related to that. Like if you have something that's in that you need to do spectral pre-processing on. So if you've got peaks of things, if you're looking at some other type of output before it gets to this data set where you've got actual numbers for expression or amount or something like that, there are some additional tools here as well under spectral preprocessing. But for the most part, you'd just be looking under expression.
Okay, so there are more questions coming in right now, but I think we will have some more time for questions later. So I would suggest to proceed with the next example. And then we will also answer all questions we don't have time to answer during the session, also offline after the session, so nothing will get lost. So no worries.
Yes. Yeah. Absolutely. Yeah. And we'll put up our emails at the end, and you're welcome to send us emails directly if there's anything that we didn't get a chance to answer. Okay, so I'm going to go a little more quickly because you've seen a little bit of JMP, and so I can be a little bit speedier on this example. So we'll spend less time here. This is an example of a genome-wide association study where we've got SNPs, so single nucleotide polymorphisms, and the samples are Bernese mountain dogs, and we're looking at a particular type of cancer. Humans get this type of cancer, but Bernese mountain dogs get it at a much higher rate. So they're a really good surrogate to explore what SNPs or variants are associated with this particular cancer. So again, I'm stealing data from online, and this is a reference to the study. And so again, I'm taking this from the GEO website. These data, they're Illumina data. I can grab the data in several different ways. One super helpful way for me with JMP Genomics is to take this minimal formatted family file. Again, JMP has tools for lots of different types of files, and there's a nice tool for this minimal file. So that's the one I'm choosing, but I could also just download the raw data, the series matrix files, whatever. So I can have a folder of all the samples, or I can grab this minimal formatted file, which is actually a little bit easier. When I do this correctly, this is what I get for the genotype information. So now we've got what's called a wide data set. We had a tall data set for expression. We have a wide data set for genetic data, where we have each sample as the rows, not the columns anymore. And now our columns are, you can see starting over here, SNP1, SNP2, SNP3. These are not labeled in a particularly scientific way. That's not a lot of information to link back to what these SNPs actually are, but we have our SNPs here, and there's 22,371 columns, so over 22,000 SNPs in the data set, and about 474 samples, 474 dogs in this data set. The genotype data in this case is sort of combined with the EDF, the experimental design file, so all the information about what was different is actually just captured in the phenotype. The breed is the same for all the dogs. The source is whole blood for all of the dogs. Many of these columns are redundant because they're just the same for everything in the data set. But the phenotype has the histiocytic carcinoma, is that what it is? Sarcoma. And it has unaffected. So we have the cancer and non-cancer. And then there's also one extra thing that we could include if we want to, and that's the origin of the dogs. Some of them are from North America, some of them are from various countries in Europe, so we could also include that if we wanted to. We, in addition, have annotation data on these. So the SNP ID is listed here. We know what the chromosome is. We know the position on the chromosome. So this extra information will let us do more things because we've got extra annotation information. So if we link this from the very beginning, it will get carried through the analysis. And then when we have just the markers we care about, we can go back and use this annotation information. So here's what the workflow journal is going to look like for this analysis. I did not use a workflow in this example. Instead, I used several different pieces and put them together. And there's another tool called this workflow builder that lets me then grab all the results and put them into a workflow as though I used a workflow. So everything lands in a journal. So all that to say, it can be really handy to sort of package up your results, even if you're doing this a la carte. You're choosing your own things to do instead of using a workflow. The first thing that I need to do here is when I bring these data in, those SNPs, I need numeric data. I have to have numeric, and I can't have missing values with something beside blank. I have to have missing values be blank. So the first thing I'm going to do is take all those NCs from the data. Those are the missing, and I'm going to replace them with blank. So I do that step. The next step, I need to not have AA, BB, AB, or any of these other codes. I need to actually have something numeric there. And so ideally, I'm going to put zeros, ones, and twos. The homozygous minor allele is going to be a 2. So whatever is the least common, but the same in both positions, is going to be a 2. Whatever is the most common and the same in both positions is going to be a 0. And the heterozygous is going to be a 1. So if you see an AB, that would turn into a 1. In this case, BB, for this particular SNP, BB is the major allele, so that gets a 0. So BB got 0, 0, 1, 0, 0, 0, 0. For a different SNP, BB might actually have been the minor allele, so it will depend on what's actually the major or minor. Okay, so we had to clean up the data, this second view here with the zeros and ones and the dots. This is what we need. Those dots are just JMP showing us that they were blank. And that's where we need to start in order to do an analysis with this in JMP. Then the next thing I look at is marker properties. I look at how frequent is the minor allele. If it's incredibly rare to have the variant, then it's not going to work well for traditional statistical methods. There are different methods for rare variant analysis. So generally, when I'm doing just a typical statistical analysis, I actually want to exclude anything that's an extremely rare variant. So I'm looking here, I'm going to use these filters. I think I have this blown up in a second. I'm going to use these filters on the side and say, where do I want to cut off? I have to have at least this % for the minor allele. So right now I've got the cutoff at 5%. So if the minor allele is less frequent than 5%, I don't include that variant to do an analysis on. Same thing with missing genotypes. If you're missing a whole bunch of the data, if this dog has 80% of the data missing, I don't want to include that dog in my sample. That makes no sense. So I can also quickly clean out things that have a lot of missingness for the genotypes. There's also a cutoff for the Hardy-Weinberg equilibrium. This doesn't apply in our case. Hardy-Weinberg equilibrium is for when you have an experimental population, not for an observational population like this, so that's not going to matter for us here. And I have a second filter for missingness, but this is for to filter out not the markers that have a lot of missingness, but the individuals. Sorry, I said that backwards before. So the top filter is for the markers that are missing. So every dog or 70% of the dogs are missing the value for that marker, then I get rid of that marker. If one dog is missing 70% of the genotypes, I get rid of that dog. When I do this cleaning, I'm going to get this little boring looking output that's still very helpful. It tells me originally I had 22,362 markers. The markers remaining are 13,684. I'm excluding 8,678, and I'm removing 10 of my individuals. So I had 474, now I'm going to have 464 dogs. And then again, boring output, but this is just links to the data sets if I wanted to go look at them, or I can just keep going from workflow to workflow and passing the data sets to the next thing. When I'm doing this variant analysis, I have a lot of options here. Most of these things are typically called GWAS, genome-wide association study, but they're sort of nested things. So a QK mixed model is kind of the biggest, the big brother statistical analysis that allows you to put a lot of extra information in the model. A simpler version of that is called a SNP trait association, and an even simpler version of that is called a case control analysis. The case control analysis is just, I have to have a binary trait. It's a very simple model, and it has to be a binary trait. So a binary trait would be that I have a case and a control. I can't have 3 levels. I can't have various progressions of a disease. It has to be yes or no. SNP trait analysis, I'm allowed to extend that. It doesn't have to be a binary trait. It could be something with many categories, or it could even be a continuous trait. Still a pretty simple model. And QK mixed models, any kind of trait, but the model now will also put in an adjustment for the population structure and the relationship between the dogs. So a case control, we're going to get the same kind of output, which is shown here on the right side. We're going to get a picture of how frequent that minor allele, major allele, and heterozygous are for the cases versus the control. So we're going to see the proportional differences, and we're going to get statistical tests on are these truly different in proportion. In this case, when we just have case versus control, we can either look at a genotype test or we can look at a trend test. So we're going to see these options in the output. If we're looking at a QK mixed model, now, in addition to just that simple, is there a difference in how expressed this was or what variant we got for the compared to the trait case control, compared to how many zeros, how many ones, and how many twos? now we can also include population structure and relatedness. So we get this Q matrix that has to do with the high-level population. We get this K matrix, often calculated through identity by descent, that has to do with the unique relationships. And you can actually see in the output that I'm looking at here, so this is also generated from JMP Genomics in the process of calculating these matrices. I've got the points, the dogs, colored by where they came from. So the blue dogs came from Denmark. This reddish color came from France. This greenish color, which we don't really see, came from Germany. The other big group is North America, is this mustard color. So the mustard North American dogs seem like they are a little bit different in principal component analysis than the Denmark and France dogs, which are more similar to each other. We see those same colors over here in the dendrogram. So there's high similarity between the North American dogs, pretty high similarity in the Denmark dogs, pretty high similarity in the French dogs, although there's a little bit of dogs being placed in different groups up here. So we're seeing that there is relatedness and similarity in their variance based on relatedness. So if we can explain that first, then when we do these tests, we're going to see, in addition to what could already be explained by relatedness, how much more does the cancer relate to these different SNPs. So if we do the QK mixed model, there is a workflow for that. And this is what the workflow journal looks like for that. We would get this PCA for population first. So that's looking at this PCA picture here. Then we would get the relatedness matrix, which is this relationship matrix on the right side. And then we would maybe do some kind of compression to make this run faster, because these are huge models that take a long time to run. And then we would actually run this mixed ANOVA model. So again, this is just closer up looking at the same things. One of the things we'll look at when we're getting those relatedness, so this relationship matrix, something else in the output that it will show us is how related the dogs are to each other. And here you can see that a few of the dogs are actually completely related to each other. So I'm finding 3 dogs that are identical in this identity by descent. So I might have found a data error here. It's possible that those are actually the same dog, because it would be incredibly rare for them to be identical otherwise. Then once I've accounted for that population and relationship structure, then I run this, again, a whole bunch of little tests, and I can see the p-values spread out. In this case, I'm spreading them across the chromosomes where those SNPs are located, so that I can just easily visually see, are there any chromosomes where I see a lot of highly significant SNPs? And I do in chromosome 11. That seems to have some of the highest, the biggest effects. So this is called a Manhattan plot. We're kind of scanning the skyline and looking for the really tall buildings. That's where we see a lot of effect, or high, really important effect. Here are 3 of our most important, most highly significant and kind of large effect SNPs. And so if I grab one of those, if I click on it, I can then ask to see that comparison between how much. So you can see that for the control, there's a lot more of the minor allele. In fact, it becomes the major allele. For the case, so the cancerous dogs, they were much less likely to have the what we're calling the homozygous minor allele. If I look at all 3 of those points, here's the comparison between all 3 of them. So if we just focus in on the 2s for this difference, you can see that there is definitely a difference between the case and control for the 2s. Here we can see that again. In this plot, it's more the 0s and 1s are what's changed. So again, using the software, we're able to filter out SNPs that have minor allele frequency, missing values. We can clean up the data pretty easily. We can then look at origin or relatedness and include those if we want to use a QK model, QK mixed model. We can use the Manhattan plot to see what chromosomes seem to be more related. We can also, again, grab what those important SNPs are and send them out to a pathway analysis to see more information on that. Okay, so briefly, let's just glance at the software for what this would look like. So I'll just show the SNP trait. So again, I'm getting tons of files here, so many files. I just usually sort by type, and then I look for that journal. So if I click on the subset and reorder genetic data, it's just going to give me a new data set, so that's not that interesting. Marker properties is where I'd have the chance to filter. So the marker properties is where I get these filters, and I can decide where I want those cutoffs to be. And once I say okay, and then I run this, then I'm going to have a smaller data set, just like I showed you. Then I would subset and reorder again, and then I would actually do that analysis. So this case control analysis is not the QK. This is a simpler analysis, but it's not going to take as long to open. So here's that Manhattan plot where we see the particular markers that were most important. Again, I can hover over them, find their label, see which SNPs these are. I can do more things to drill down and search for these in other databases. And I can look at this by chromosome. So there's a bunch of sort of different ways of viewing this. Okay, so any questions on GWAS type of analysis? So I'm just going to give you a very brief overview of what you could do with plant breeding. This particular example is, I think we've got a few documents on our website that show this example, and certainly if you're doing this and you want more detail, ask me about it. I'd be happy to send you some helpful documents that would sort of walk you through an example of how to do this. I believe there's also a video on the JMP website in the Mastering JMP videos. If you're interested in this, let us know, and I can send you a link to that as well that explores this in a little more depth. But basically, what we want to do is, again, clean the data a little bit, just like we did for the genetic data. And then we want to fit a predictive model for the phenotypes, and then we want to simulate the progeny. So the idea is, what I have here are plants where I have 5 different traits, 5 different phenotypic traits that I'm interested in. I think plant yield is that PY variable. Pro is a measure of protein. I think SSW is seed weight. And I can't remember what the other 2 are. So what we're going to do is try to maximize or minimize or whatever it is that we want to do with these traits. And then, so we'll build a model that predicts from the genotypes what the trait would be. And then we'll maximize or minimize to choose the best things to cross. Then we'll cross them, and then we'll keep checking each generation which ones we want to switch out. And then we'll, in the end, see what we think happens generations down the line after we've carefully predicted and crossed these guys. So this is the kind of stuff we're going to be looking at. These scatter plots at the bottom, what we're looking at, the scatter plots correspond to this list on the top. So the first thing we're seeing is the ERG mean, then the ERG standard deviation, then max, and then min. So we can look at different things besides just the mean, but the mean might be the most important one. So if we look at the mean of ERG versus the mean of SSW, so that would be like this plot right here. We can see the general distribution of when plants have high ERG and high SSW, then they would be sort of in this little tiny region at the top. So what we want to do is use these sliders on the side to say, hey, let's try to maximize the ERG average. Let's try to also maximize SSW, but I'm going to give it more range. This one matters more to me. I want to really get to the maximum of ERG, but I don't care quite as much about SSW. I don't care anything about TW or the protein. But then the plant yield, I also want to be pretty high. So we can play around with these sliders. That's one option of how we can choose the regions that we care about. And as we play around with those sliders, then we're starting to zero in on exactly which genotypes are responsible for these predicted high values. So this is zooming in a little closer on these sliders. It gives us the minimum and maximum that were predicted, and we just sort of slide around to the region that we care about. Then as we explore these, we can start zeroing in. So we've chosen already some of our favorite things. Now we can start zeroing in on what would happen when we cross them. So this line, NOS 161 crossed with NOS 62, this is its predicted simulated mean. So this is if what we wanted to do was maximize that ERG, this is a really nice candidate. Crossing 161 with 62 is a really good idea. Likewise, this one is a really good one. This is crossing 62 with 64. So interesting that 62 falls in both of these crosses. So I definitely want 62 to show up in my crosses, and I might want to cross with 64 and 161. So we're going to look at these points and sort of pick out things and zero in, and then each generation we can do this interactively, or we can do this by just setting the computer to do it. If I set the computer to do it or do it interactively, I can then get what I call a caterpillar plot, which is showing the first generation, the second generation, the third generation, the fourth generation. So I can see that for ERG, my predictive model was really strong, and I actually am really able to control this. There was a strong relationship between the genotypes and the ERG, and that strong relationship lets me actually make a difference as I make these crosses. So you can see some of them are a little bit less straight, but all of these are moving in that positive direction the way I want them to, except maybe this guy here. So here I'm able to see that improvement I make generation by generation. We won't talk about how to do this in JMP Genomics. I do want to show you quickly in the software the predictive modeling tools that we have. So what we would start with is under predictive modeling. And what I really like is this thing called the Predictive Modeling Review, which lets me choose lots of models at the same time. So here's all the models that are available: a distance scoring, general linear models, partial least squares, partition trees, quantile regression, radial basis machines, ridge regression, discriminant analysis, k-nearest neighbors, logistic regression, a bunch of things. Genomic BLUP is a very common thing to use when you're doing plant breeding. So this is very likely going to be in the mix, but I might try other things as well. Ridge regression often works well in plant breeding contexts. If I choose the Predictive Modeling Review, I think I clicked on that. Why aren't you responding? I must have something else open. If I click on that Predictive Modeling Review and it opens for me, you'll see a way to choose as many options as you want. And there's a bunch of internal filters that you can use as well. So a lot of things to do. Yeah, here we go. So if I click on the models, I can choose from all of these models and I can edit each model. And there are a bunch of options for, like, if I want to force some of my SNPs to be included, I can lock them in. If I want to reduce the number of predictors, I can say just pick the 500 best. Don't pick all 23,000, just pick the 500 best and do it by a regression or a forest, or what method do I want to use for predictor reduction. So there's tons of stuff in here. It's really deep, and it's a wonderful tool to use to do in silico breeding. Okay, so we are 2 minutes over. I'm happy to take questions if anyone is still here and has questions for me right now, but I'm glad you were able to join, and we'd love to help you if there's anything we can help you with in the future.
Great, Ruth. So just a few questions. Here's the first. What equation or prediction is the software using to predict the phenotypes of the progeny?
Yeah, that's going to be just whatever models you're choosing here. So if you choose like a ridge regression, it's going to use that methodology to do the prediction. So you'll build the predictive model and then you'll use that for the progeny simulation.
Okay. And the second is, will you be able to elaborate a little more on the linkage disequilibrium analysis?
Sure. So I actually think probably the best thing is there is a document on, I think there are a couple places we can find this. Probably the best place to look first is at community.jump.com. This is our user community. There's a nice set of blogs on how to do some of these things in JMP Genomics, and I think there's one on linkage disequilibrium there. So I'm going to go to community.jump.com, and everything on my computer is just very slow right now. I'm going to search for JMP Genomics. It is a blog, so I could search in that area, but I think this tag will be sufficient. It's not this Bayesian add-in, it's not this quasar alignment add-in, it's not this outcross thing, it's not this big, here we go, genetic association with JMP Genomics. I think it's included in this. So I'm going to click on this. And here are a bunch of tools, a bunch of different things that you can get instructions on. So if you need help importing genetic data, if you need help with the basic genetic workflow, with that case control kind of stuff I showed you, or with QK mixed models, here's linkage mapping and QTL. So I think this is, so the basic linkage mapping is probably what you're asking for, or QTL, single marker analysis, would be related to that. So I think this is probably where you want to go. So again, I just went to community.jump.com. I just searched JMP Genomics, and I found this series called Genetic Association with Jump Genomics. There is also a page called jump.com/jumpg for Jump Genomics. And this page has a few other documents. Some of it has a lot of overlap with what's in those blogs. So there are some step guides. So I think there might be one here on maybe under the additional step guide. So I don't see it here. So the blog may be the best place. Those are good resources for anyone who has more questions. So feel free to community.jump.com and search Jump Genomics, or jump.com/jumpg.
Ruth, there was one more specific question for estrogen treatment, dataset microarray.
Yes.
We have to import CEL files? Question mark.
You do not have to. So that is one option that's available. So under the import, there are lots of types of files that you could import. So under Affymetrix for cell files, there are different import engines to help you import cell files and different kinds of files. If you have Illumina data, there are import engines to help you with that. If you just have text or CSV or Excel files, that's also easy to bring into genomics. If you've got other types of expression data, you might have something falls into these categories. If you have next-gen sequencing, you might have SAM or BAM files or read counts, bin intensities, VCF files, so on. So there's lots of different types of files that you could bring into Jump Genomics. It's not always incredibly straightforward, depending on how the files are themselves arranged, but we've tried our best to be aware of what are common file types and to build importers around those. And if you're stuck, we have wonderful tech support, and I love to help people through things. So let us know if you get stuck on the data import. That's the trickiest part. Once data's in Jump, you're kind of just walking through workflows, but lots of things can mess you up on the import depending on what might have changed in an output file from another software.
And for SNP, which kind of data do we have to input?
Again, text files is always easy. You might have imputed SNPs in different formats. You might have data coming from any of these software packages. In my experience, I feel like I'm very often reading in text or CSV or Excel files for a lot of things, but that may be because I'm usually getting them secondhand, not getting them straight from the software, from other software. So again, if you don't find a tool for what you're looking for, send us a question and we will be able to help you figure out if your particular data set can be read in. FASTQ files cannot be read in. If you're doing alignment, Jump Genomics will not do the alignment. We need the data after it's been aligned. So for expression, we would not be taking the FASTQ files.
Okay, so there are even more questions coming in, and it's very nice to see that some participants seem to be already some Jump or genomics users.
Great.
And they also started to share some tips and best practices. So really, thanks a lot for engaging with us and for joining us today. Maybe some people asked about the blog. Maybe last but not least, we take this pointer and show you where to find the genomics.
Yeah, as Volker said, we can send you a link to this as well, but on our community.jump.com, there are a number of people at Jump write blogs, and even people outside of Jump who just love Jump will write blogs for us. So you could go to the Jump blogs or just search from that very first search page, search Jump Genomics, and you'll find this set of blogs about expression analysis, a set of blogs about genetic association analysis, and you'll find a whole bunch of tips and pointers. This is also a great place if you are a Jump user and you've got tips and pointers for other users. If you want to write comments on any of these to say, hey, here's a really easy way to do this, or if you're stuck on this step, here's something to do. That stuff is really useful. We'd love to have our Jump Genomics users interacting with each other as well, because often you guys are seeing things before we are.
Okay, so as you saw, this is really JMP Genomics is a SaaS JMP product. It's a real product. It comes with a full set of documentation and supporting material, technical support, and also the academic team is there if you have any questions about the product itself or getting a license. Please don't hesitate to get in touch. And if you want to join the boat, and there are so many applications and use cases, and we just saw a few of them today, that there's so much to discover. If you want to join the boat, you are very, very welcome.